So I'm going to talk to you about reproducibility today. And so I want to say, just to start with, that this is the first time I give a lecture on that subject. So I also had to, of course, do some significant research to decide what I want to cover today in this lecture. And what I found is it's actually a very fascinating topic. So the more I digged into the research and the literature about reproducibility issues, it's really interesting, and I hope I can convey some of this to you today in this lecture. I have actually one slide to introduce myself to you again. So I'm going to start in just a few weeks my own research group at the Max Planck Institute for Infection Biology, and my lab will run under the title Innate Immune Regulations. So what we study are immune receptors in the context of the cellular architecture. So my system is interested in autolytic receptors that are sitting in endosomes, and they sense nucleic acids. And so some of the questions we try to address is, where exactly do these endosomal TLRs get activated, and how does the localization of these receptors dictate their response behavior? So if you think this is cool and you would be interested in that, we start hiring beginning of next year. At all levels, basically, PhDs, postdoc, master thesis, internships, whatever. So you're very welcome to contact me. The outline of my talk today, so I'm going to split it up in two parts. So I want to talk a little bit more about the replication crisis. We've heard a little bit already today in the morning. Its origins and also its reasons. Why are we in this crisis? And then factors that lead to poor reproducibility in science. So basically, we start out with the problems, and then we want to move from the problems to solutions. So of course, this crisis had led to a counter movement or a countermeasure, the open science reform movement, that tries to improve reproducibility by revising, publishing, and open data policies. And I also want to give you some practical tips for publishing in this second part of my talk. And also, I want to do two quizzes throughout the talk using Mentimeter, so please keep your smartphones close. I will call you up, and then you will go to menti.com and put in a code so that you can participate in the quiz. So I'll tell you when we're doing that. So I want to start out just very briefly to have everyone on the same page. What do we mean by reproducibility? Because it can actually mean two things. And for the sake of the talk, when I talk about reproducibility, I mean these two things together. So it can first mean that a study can be basically carried out again, assuming that methods and the procedures are explained in sufficient detail. But it also means that the results, that the replicate study can successfully produce the same or sufficiently similar results as the original study. So when we talk about reproducibility, I mean both of these points at the same time. So we are facing a replication crisis. And so this all began about 10 years ago. And what happened is that pharmaceutical companies, primarily in the US, they went public and claimed that more and more scientific results cannot be reproduced. And this was a huge problem because it meant that a lot of money has been wasted by these companies. They move on to perform clinical trials with compounds that turn out to be not effective. And furthermore, it's also very unethical towards the patients because they're patients hoping for new therapeutics, for these clinical trials to be successful because they might be able to help them. Clinical trials in oncology specifically have the highest failure rate. And I think this is because cancer is it's a very prevalent disease, and the cancer research field is also very fast progressing. So there's a really high pressure to come up with new, innovative, and successful anti-cancer therapeutics. So that also plays in why you find this issue, particularly in the field of oncology, very prevalent. And so you might say now, or you might ask yourself, so why do we care about this? And so the reason is that a lack of reproducibility could ruin the reputation of basic science, actually. So if scientific publications cannot be trusted anymore, they can no longer be taken at face value. This is also a problem that directly relates to us as the scientists. And it also means that publications that are not reproducible, they trigger a series of follow-up studies on wrong fundamental bases. So it's actually perpetuating itself. And if society loses trust in science, this could, of course, also result in funding cuts, because we have to remember that most of our funding is actually coming from tax money. And so there are countermeasures that came out from this crisis that now resulted in much stricter publishing guidelines. And this is also affecting us because we have to put now, in order to improve this, we have to put in more work for publishing and for correctly documenting our research when we write up our manuscripts and papers. So the first question I want to ask you, just to get an idea of how many of you have actually heard before of this replication crisis. Everyone? Wow, almost everyone. That's good. I still see counts coming in, so people are still voting. Okay, we give it a few more seconds, but it seems like 74% of people have heard of it before. So we move on to the next question now. So how bad is it? Okay, for the second question, how many publications can be successfully replicated? What do you think? A lot of pessimists, yeah, 10 to 20%. We'll stop here. Thank you so much. So it's actually, the right answer is actually 10 to 20%. Only 10 to 20% of papers that are trying to be replicated actually can be replicated. So I want to tell you a story of like two big replication studies so that you have some examples. One of the first ones that had been carried out was from a big pharmaceutical giant in California called Amgen. And what Amgen tried to do, this is more than 10 years ago, they tried to confirm 53 landmark preclinical cancer studies. And it turned out that only 6 of those could be reproduced by Amgen, so about 11%, which is really shockingly low. One drawback of this study was that Amgen has never identified the actual papers they tried to replicate. So they also never contacted, or they were not in exchange with the original authors while they were replicating them. And they also never published really detailed comparisons because they wanted to protect the original authors. So you can actually not go and look what exactly they have done and what their exact results were. But there was another study that's also quite famous, the Reproducibility Project in Cancer Biology, that was carried out a few years later by the Center for Open Science, which is a non-profit organization also based in the US. And they attempted to replicate 50 of the highest impact cancer biology studies published between 2010 and 12. And recently they notified they will only complete with 18 replication studies, so 18 out of these papers, because too few originals reported enough information to proceed with full replication. So this is just like pointing out that really in a lot of papers that there's not enough sufficient documentation that these studies can actually be replicated. And the results were reportedly mixed. So 10 of these studies have been published in eLife in 2017. So you can go back and you can look those up. So this study actually published and identified the original papers and the original authors. And of course, this ended up like in a huge dispute. Of course, original authors felt jeopardized, their careers, their reputation felt jeopardized. They were saying, they were refuting, of course, the results of the replication study. They were saying that this is their results, these were their findings, and many other labs have reproduced these findings so far, and now there is this replication study which cannot reproduce the results. So what does this actually mean? It's basically now 1 against 1. And so it's just to show that it's not so easy to prove who is actually right and who is wrong here, and that these replication studies can also like induce a lot of disputes and like heated conversation. And basically the Center for Open Science basically stated then just the inability to replicate a study does not mean that the study was wrong to begin with. So to phrase it softer, but it's a complicated issue. So I want to cover a few points, what the crisis refers to when we talk about the replication crisis. And I'm going to go over this list rather quickly, because then we will go into detail in some of these points. So the crisis often refers to the virtual absence of replication studies in the published literature. And this is basically a fact. So if you go and you look through our literature, scientific literature, you will barely find any replication studies. Like everything you find is new, because this is what journals want to publish. They want to publish novel, new studies. And of course, also the widespread failure to reproduce results. So we talked about this. I've shown you these two examples of big replication studies that had rather shocking or sad results. The main reason for why we are in this crisis is basically the publication bias. And so this is under publication bias, and I will talk about this more in the next slide. We understand that the journals only want to publish positive results, significant results. And so this leads to a high prevalence of questionable research practices that scientists engage in, which inflate the rate of false positives. And this, in turn, increases the lack of reproducibility. And also the documented lack of transparency and completeness in the reporting of methods. So we need better ways and guidelines to better document what we did exactly and how we analyzed our data and how we came to our conclusions. So what's the publication bias? So many scientific journals had historically explicit policies against publishing replication studies. So there are many examples where they simply rejected a replication study, saying that's not new, that's not novel, so we are not interested in it. And likewise, our science funding bodies, like they also only want to fund research that's novel, original, and or groundbreaking in its nature. So there's a very strong bias towards statistically significant or positive results. So when you look through papers, when you look through the literature, you'll notice that about more than 90% of the data pieces that are presented in a manuscript are statistically significant data pieces. And this creates a problem that's also called the file drawer problem. So all the non-significant results, they move into a hidden file drawer, and they're basically never talked about. They're hidden from the public view, and they're often not included, not talked about in manuscripts. This also means that we never learn about them. And so in labs, many people, scientists, repeat the same experiment over and over again because they didn't know that this experiment had been done in the past, but just turned out to give no significant results. So this pressure basically to obtain significant results leads to questionable research practices that scientists engage in. So this basically means bringing non-significant data back to significance so that they can be published. So it's basically tweaking your data. And there are three main ways how this can be done. One of them is p-hacking. I'll explain this in a second. Then there's cherry picking, and then there's HARKing. Those are the three most prevalent questionable research practices. And this is not, don't get this confused with fraud. So they are not considered fraud. They're like in the gray zone, they're considered questionable. But we still, of course, should not engage in this behavior because they really inflate the false positive rate in papers and leading to lower rate of reproducible results. Another thing that's very often in the biological research is you find that the average statistical power is often too low. That also starts in the study design. So it basically means that we on average, use too low sample sizes in general to really have a high confidence that the effects we see, that they're actually true. So this is something to consider when you already think about your experiments, like how often would you have to reproduce it and how often to repeat it to be of high confidence that your result is true. So p-hacking is one of the questionable research practices. It's also called selective reporting. And I want to give you a few examples of p-hacking. So one example would be that you look at your data and it just doesn't quite reach statistical significance. And you decide, well, okay, I'm just going to add a few more data points and then we see if it's significant. This is already p-hacking. Or the reverse, you have a data piece where you have statistical significance, but you didn't collect a lot of data, maybe just 4 data points, but it is statistical significance, so you stop collecting more data to not ruin the statistics. That's also p-hacking. If you exclude data points, for example, outliers, to make the p-value look even smaller, even better, after checking the significance first, that's p-hacking. And also adjusting statistical methods or models. So you choose the type of statistic that gives you the smallest p-value. That's also not okay. And then I've seen cases rounding of a p-value. So there are reports that scientists just round up the p-value or round it down from 0.053 to 0.05, and then it's considered statistically significant. That's also not okay. So cherry picking refers to practices where it's basically like you're choosing the best picture or you're choosing your best example out of a series of experiments. Or it could also be that you don't report certain variables that did not reach statistical significance. So for example, let's say you want to characterize an inflammatory phenotype in the mouse, and you look at different cytokines, and for TNF it looks great, you have a statistical significant difference. IL-1 looks great, but IL-6 actually shows no effect. And so you just decide, well, then just not talk about IL-6, because not to complicate the story. So you just leave out parts that do not fit and do not reach statistical significance. And HARKing is another one of the questionable research practices. It stands for hypothesizing after the results are known. So basically, you obtain an unexpected result and you present it as having been hypothesized this from the start. So which actually means that you're twisting the story. And this basically twisting the story conceals a useful part of the truth, and that's why it's also unethical to engage in this type of practices. So here is just like a graphical summary of all these questionable research practices and where they come to play during the scientific process. So when you start out, you start out with an idea and a specific hypothesis you want to test. Like in general, most of the scientific work is usually not replicated. So there are basically no replication studies found. So this is one of the things. Then you start your study design. So how do you want to address your hypothesis? Here often the statistical power is too low to really be confident that the effects you will find that they are true. You start collecting your data, you analyze your data, this is where p-hacking happens, and you interpret your data, this is where p-hacking continues to happen. And then when you have unexpected results that are really cool, you engage in HARKing, that just you twist the story and you say, oh, I've hypothesized that actually from the start. This is what I was expecting to get. And when you publish, there is, of course, you are a victim of the publication bias. You only publish your positive results. And often there is a lack of data sharing or not enough information shared concerning the methods and the procedures. So I want to do the second quiz now, the prevalence of questionable research practices. So how often do you think do scientists engage in these practices? And so I'm switching back. Okay, so the first question to you is, which questionable research practice is the most common one? Yeah, it's looking really good. So it's actually, that's correct. Cherry picking is the questionable research practice that's most commonly engaged in, followed by p-hacking and then HARKing. And these numbers are actually from biology studies, not exactly our field, but there have been surveys conducted for ecology and evolution biologists. And these are numbers that are coming from these studies. So the next question, how many researchers do you think have at least once engaged in cherry picking? And again, this is based on surveys. So people basically self-report in the surveys. So the actual numbers can be either higher, because of course not everyone is going to be fully honest, like self-reporting himself, even though these surveys are anonymous. Okay. So that's also correct. So more than 60% of people, scientists in the biology field, have engaged at least once in cherry picking. And I'm pretty sure the reality, it's going to be much more. It's going to be close to 100%. It's also, cherry picking is also very prevalent in psychology, in psychology sciences. There it's almost 100% that have been reported. So I'm going to go back to my presentation. So we want to talk about solutions now. So out of this crisis, there was a movement formed that's called the Open Science Reform Movement, that aims to improve reproducibility in science by several measures, like by data sharing, for example, by improved documentation, register reports. I want to talk about this is a new form of publishing a paper, a new form of paper, and also stricter editorial policies around statistical reporting. And this includes also stricter editorial policies, including publishing replication studies. So more and more journals are now welcome to publish replication studies and to publish statistically non-significant results, to take out this publication bias, to at least reduce it. So what do these points here, what do they mean for every one of us, also like in practical terms? So data sharing, this means now when you publish your paper these days now, you will have to make much more data available when you submit than you used to do in the past. So now journals might ask you to submit all the raw data, basically, that accompany the figures. They're also called source files, raw data. You may be asked to submit metadata or code, and you can either upload them into open source repositories or databases, or you submit them as extended files to the journal directly. And even fellowships may ask you, or funding agencies, to participate now in open research data plans. I know, for example, that Marie Curie is doing that on a voluntary basis for now, but also they will ask you to come up with a plan how you want to share your data when your project is completed and when your project is being published. So just to be aware, it also means to you there's much more workload on you. Now, when you put your paper together, please organize your data in a smart way from the beginning, because it can be really painful to go back and dig out all this raw data when you don't have it together with your actual figures, and then journals will ask for it. So I want to give you some actual examples. So this is a figure from a paper, like a very typical, this could be the Western blot panel, or it could be gel bands. And so if you submit this, nowadays, this is not going to be enough. So the journal will ask you for the raw data files, for the source files of each of these cropped gel pictures. They will ask you to provide the uncropped raw images that contain the letter. They want to have the letter labeled and to contain the loading controls. So just imagine how much work this is going to be. If you present a panel like this, it's going to be for sure a full day of work just to get all this raw data and prepare this to be able to send it, to submit it as an extended source file to the journal. So you better have your data really well organized. A second example, if you would publish a figure like this, like a dot plot, and here a facts plot, and here a bar graph, the journal might ask you now to provide for every graph an Excel table with the actual numerical values. So you have to do this on the side for each graph that's presented in your figure panel. And you might be asked to provide every single repeat of an experiment if you just showed a representative experiment in your figure. So they want to see all your background experiments, repeat experiments, from where you only cherry pick the one representative to show. And then for facts plots, they might ask you to show the entire gating strategy, starting from forward side scatter all the way to the final gate you're showing in the figure. So just be aware that this is also more work to do. So have that ready. Know exactly how your gating strategy works, because they might find errors basically when they look through your gating strategy and something doesn't look good. This is not, of course, this slows down your publication. They might ask you to include fluorophores and antigens, so be really thorough. Also, another thing is they might ask you for lot numbers. This never happened to me before, but it's always safer to start documenting lot numbers in case there is some discrepancy or something has to be evaluated or dig further in. One thing that happened to me personally is, so we had, the paper was written, it was accepted, and then we had to click that button, test the authenticity of your cell lines, like make sure, proof that these cell lines are actually what you think they are. And so the journal wanted to know whether we had done mycoplasma testing, whether they were free of mycoplasma, and whether they were authentic. So we had to send those cell lines in after the fact that the paper was written and ready to go. And we sent them in for STR-DNA profiling. And it turned out, very lucky for us, that they were authentic. But now imagine if the HeLa cells you worked with are only 60% HeLa cells and 40% contaminant. That would be problematic at the point where the journal actually already accepted your paper, and now it turns out that your cell lines were not clean. So many journals will ask you for this now. So be really aware the material you work with, that this is the right material. So there is this new form of papers. I just learned about it. It's called registered reports. And registered reports, they try to eliminate the publication bias. So they try to eliminate the prevalence of questionable research practices. And the way they do that is, so you would develop your idea, and then you would write up a study design. So your hypothesis and how you want to test it. And then you submit this to stage 1 peer review. And experts will look at it. And if they like the idea and if they think the study design makes sense, they will give you green light, and then you start to do your experiments. You start collecting your data. And no matter what results you obtain, so whether your hypothesis turns out to be true, whether your effect is significant or there's actually no effect, the journal basically guarantees that your paper is going to be published, no matter what the results are. So you still have to write it up, of course, your paper, and send it in for stage 2 review. And they would basically look whether you stick to your original plan and then publish. So it's actually, in a way, it's much more safer. It's much more honest. Like you really present, okay, we had this hypothesis, but there was no effect. This is what it was, and it's still gonna be published. So this is a rather new idea. There are not a lot of register reports out yet that you can look what type of studies would that be. I looked up which journals would participate and would offer this format. And right now, there are only 3 that would be maybe most relevant to like biology or like immunology. This would be Nature Communications, just started to offer this format. So they are taking registered reports and follow this format. PLOS Biology does that, and PLOS One. So it's not a lot of journals, I have to say. So I'm hoping that with time, more and more journals would participate in that format. Maybe even high-end, high-impact journals, we have to see. but I think it's a very attractive way in publishing. So I think I want to try this out for sure, at least once, to see how that goes. Of course, at the beginning, it's a lot of work to put in because you have to really thoroughly explain what you want to do. But then it's basically a very safe way, and you take out this component of luck, basically. So no matter what you find, you will have a published result at the end. And so I want to talk at the end a little bit about statistical reporting and what changed here in terms of what the journals implemented for changes. And I want to recommend here an article comment from David Vokes, who published in Nature. So you can find this article also in the publishing guidelines of Nature. So David urges, so it's called Know When Your Numbers Are Significant. And David urges basically experimental biologists, reviewers, and publishers to grasp basic statistics, because otherwise sloppy science will continue to grow. And so he gives a few recommendations in this comment article, and three of them are that I pulled out for you. So what David asks for is figures with error bars in your manuscript. They should always state what these error bars actually describe. So often you find error bars and it's not clear what they stand for. Like, is this error bars from independent experiments? Are these error bars from just technical repeats? And so he goes on and says error bars and p-values for single representative experiments make actually no sense. So even if you have replicates in this experiment and you show 5 data points, if these 5 data points have been recorded in the same experiment, it's basically an N of 1. You've only done this experiment one time and you're just showing this one experiment. That's not representative for the whole population. So statistics and error bars should be only used for independent data and not for identical replicates within a single experiment. So you see already he distinguishes between independent data and replicate data. And so real independent data means you obtain values from separate independent experiments. So you would do an experiment with a new batch of cells. You would stimulate them with a new batch of ligand, and then you would read it out on a different day. Versus replicate data is basically you have several replicates within the same experiment. And this is actually just an internal check of your own performance or of the experiment's performance. Basically, how well is your pipetting going in this experiment? It's not really reflecting the population variation. So, of course, so now if we should combine independent data into one graph, sometimes that's actually not so easy. So maybe some of you have encountered that already. Sometimes independent experiments, they're not easy to put in the same graph because the data points just don't match up so well. And so I want to give you one example, an easy way of how you can normalize your data, independent data, when you want to show them actually in the same graph. And this is a normalization method I came first across in a book about designing animal experiments. So let's assume an example. Let's assume you want to measure a concentration from a control group and 2 treatment groups. So this could be, for example, an ELISA to measure a cytokine concentration. And you perform 3 independent experiments. And for each experiment, you have 3 replicates of each of these conditions. So you end up with 9 data points. And so now you plot these 9 data points in a graph. And this doesn't look so great. I mean, for maybe here for the first comparison, you get 1-star significance, okay, but for the other one, it's not significant. And it's just all over the place, these dots. So when you stratify your data now and you look at the individual experiments you did on 3 individual days, you'll actually notice that the trends were actually always the same. So your treatment 1 was always lower than the control, and your treatment 2 always higher. So your trend is there, but just when you assemble all these data points together, it doesn't look that good because the variation between independent days is just greater than the variation between replicates. So what can you do? And it's a very simple way. It's called normalization to average. So what you can do to make this look better is you take every data point and you simply subtract the experimental average. So you would take the average of the 3 orange dots, would subtract it from each orange dot. You would do this for the blue dots and for your third experiment. And then you add back the grand average, the average of all the data points. And when you do this for every data point, you normalize to average. And then you can normalize and get a graph like that. So now this makes a lot more sense. But be aware, when you normalize data, you're not showing your actual values anymore on the y-axis. So now it's normalized concentration. So this is the drawback. When you normalize, you lose your actual raw data, but it's a way to like combine your data. You could, for example, show this in a figure, and then in your supplementals or in your additional source files, you can show the individual repeats so that reviewers can follow what you did. And you always have to explain very carefully how you normalized, what you did with the data. But that's just one way I thought this is very useful. Then another point that David Folks made in his comment article that I found interesting was that there are typically two types of publications in biology. There are the type of publications that include large data sets, so usually like omics type of experiments, for which there are a good number of statistical tests and procedures explained, that there are standards for statistics. So this would be, for example, epidemiology studies, psychology, clinical trials, genome-wide association studies. But then there are also these other type of studies, such as cellular molecular biology, biochemistry, and classical genetics that do not produce large data sets or use omic type of studies. So, but what's nice to consider about these disciplines is that often what cellular molecular biologists do is they have the luxury to probe the experimental system in multiple independent ways. So usually you have a question and you address it in like multiple ways to conclude something from it. So by doing this, you often get away with smaller sample numbers. That's okay then, because if your different results that you obtain from different directions are coherent and make sense, then you can make conclusions from them. And so then you don't need sophisticated statistics. Then it might be enough, like the example I showed you before, to normalize to average and then run a t-test, and this is enough if you do several lines of investigation to prove your point. And so with this, I'm at the end of my talk, and I want to just have a last future outlook. So covering a few small points. So basically, I hope that you could see that solving the replication crisis in science basically requires fundamental changes in science organization and the entire evaluation system. So it's a rather complex issue. And so some of these changes, for example, are to enforce the rules of good scientific practice. And this is actually what we're doing today here, by making people aware, by talking about these issues, to improve in the future, like reduce fraud, fabrication, reduce the lack of reproducibility, and also to increase sanctions against misconduct. So not only towards students and young investigators, but also towards senior group leaders and professors, like everyone who engages in misconduct. And on the contrary, also reward good behavior. So there are some systems where you can earn badges or like quest rewards if you continuously share your data publicly. So there are also incentives like that. And we also need new criteria to assess science. So some of this would be to promote reliability and reproducibility, as well as curiosity-driven science, instead of short-lived sensationalist research. So I think that this new format of papers, registered reports, are trying to work in this direction. And we also need to reinforce the values of community and true scientific collaboration, and reduce this enormous competitiveness. So there's an enormous pressure on scientists that is really driving them to engage in questionable research practices, to engage in fraud and fabrication. So this has to change from within, to have just more mutual approaches. And also another thing that journals started, and we talked about this today, is also more transparency in the academic evaluation. So now you will see that more and more journals will also publish the reviews. alongside with the manuscript, or even identify the reviewers by names. And so by doing this, usually when reviewers are identified by names, the reviews are going to be much nicer. So that the criticism is going to be much more constructive and not as destructive, because they're going to be identified and published. So this can also uncover hidden networks of science by making names of peer reviewers public. So that's another way that journals try to improve basically this crisis and get out of it. And so, yeah, I hope this, and so not every one of you is depressed now at the end of this talk. I hope that actually that you can see that the scientific community tries very hard to find solutions for this problem and to improve the scientific inquiry, so that it's for all of us a more enjoyable and a more honest and a more fair experience. And so I think that everyone can do a part here, every one of us, to like improve and make science basically a better place. And so I would like to end here, and I would like to thank you for your attention, and I'm happy to take questions. And here's also my email in case you want to email me later on with a question. I'm happy to answer it. Thank you.